What makes an antibody able to deal with bacteria, or an enzyme able to speed up reactions in our body? Basically, what gives proteins their function? Well, the answer is quite simple. Structure and function always go hand in hand. One of the most important things that define a protein and determine what it does is its structure. This is a fundamental underlying principle in all of biology, and understanding this will make it so much easier to grasp the countless roles proteins and molecules in general play in our bodies. Conformation is the term used to describe the way a protein's atoms are arranged in space. A single protein can have multiple conformations, meaning different ways its atoms can be arranged without breaking its covalent bonds. But here's the thing, only one or at most a few of these conformations actually allow the protein to do its job. When a protein is in this functional shape, we call it native. You can think of protein structure as a step-by-step folding process, like a hierarchy where each level depends on the one before it. It starts with the primary structure, which is another way to call the linear amino acid sequence, and builds up through the secondary structure, in which this linear sequence gains specific shapes, like alpha helices or beta sheets, tertiary structure, in which the polypeptide folds into its native conformation, and finally quaternary structure, in which two or more polypeptides bond together. So let's break it all down. The primary structure of a protein is its linear sequence of amino acids, the fundamental building block of everything that follows. For example, the diagram here shows the primary structure of insulin, which is made of two subunits, and fun fact, it was the first protein to ever be fully sequenced. Since the amino acid chain largely determines how a protein will fold, it's worth very briefly noting where the sequence itself comes from. It's encoded in genes. DNA carries the instructions which, through a process called transcription, lead to the synthesis of a specific type of RNA called messenger RNA. This mRNA, carrying the genetic information, is then transported to the ribosomal complex, the molecular machine responsible for the translation of the genetic information and the building of the amino acid chain. The sequence of nucleotides in the mRNA directly determines the sequence of amino acids in the resulting protein. Even though we can't fully predict a protein's function just from its amino acid sequence, scientists have noticed that similar sequences tend to produce proteins with similar functions and structures, and that's why we classify proteins into families. The secondary structure is how nearby amino acids organize themselves into recurring patterns. The two most important structures are the alpha helix and the beta sheet. In the alpha helix, the polypeptide backbone coils into a spiral with R groups or side chains sticking outwards. Each turn of the helix contains 3.6 amino acids, and this structure is stabilized by hydrogen bonds between the NH group of one peptide bond and the CO group of the amino acids 4 positions ahead. In the beta sheets, instead of coiling, the polypeptide forms a zigzag pattern, creating sheets. These are held together by hydrogen bonds, but unlike in alpha helices, where bonds form within the same strand, beta sheets can form between adjacent polypeptides intermolecularly. Both of these structures help stabilize the protein while allowing flexibility where needed. The tertiary structure is the full three-dimensional shape of a single polypeptide chain. Here, we are talking about how amino acids far apart in the sequence interact to create the final fold, and you can notice alpha helices and beta sheets interacting with each other. What determines the tertiary structure is the R groups, that is to say, the variable parts of each amino acid. The sulfate bridges form between cysteine residues, locking parts of the protein together, while non-covalent interactions play a big role too. Hydrophobic interactions cause water-repelling non-polar side chains to hide inside the protein. Ionic bonds cause positively and negatively charged groups to attract or repel each other. And hydrogen bonds and van der Waals forces help fine-tune the final shape. Since tertiary structure depends on side chain interactions, it's unique for every protein, and not as predictable as secondary structure. As the protein folds, it settles into its most stable and functional shape, its native conformation. Now, if I take, for example, one myoglobin in its folded and functional form, and then add 3 more myoglobins, I get this polypeptide complex called hemoglobin, one fundamental constituent of red blood cells. It is made of 4 subunits, 2 alpha and 2 beta chains. The bonds holding this together include hydrophobic interactions and ionic and hydrogen bonds. Some proteins don't work alone. They form complexes made of multiple polypeptide subunits, and this is where quaternary structure comes in. It describes how different polypeptides assemble into a functional unit, and you can visualize the repeating patterns in the chain pretty clearly in these images. So, why should you care about protein structure? Well, because structure determines function, whether a protein is an enzyme, a hormone, or a structural component. Its role is directly linked to its shape. If a protein loses its shape, it loses its function. This is what happens in diseases caused by protein misfolding, like Alzheimer's. The ability to design and modify proteins is crucial in fields like medicine and biotechnology. So, next time you hear about proteins, remember, it's not just about what they're made of, but how they fold and interact. I hope this was helpful. Please like and subscribe, and happy studying!